welcome back. Today we are going to discuss about the topic multiple allele. Okay, before going into the topic, uh, we'll know what actually multiple allele. Multiple allele generally means occurrence of more than two alleles for a single gene is known as multiple allele. What it is known? Occurrence of more than two alleles for a single gene is said to be this type of condition is said to be multiple allele. What are the examples of multiple alleles means? Blood group of humans. Second one is fur coat of rabbits. And third one is self incompatibility in plants. As we are plant breeders, we'll mainly focus in this self incompatibility in plants. Before that, we'll like some glance about these first two also. There are three blood groups. The three blood groups A, B, and O, right? The A will be coming like IA, IA, or IA, IO. B will be right IB, 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 IO, where O blood group will come like IO and YO. If these alleles are there, these respective blood groups will be coming, right? This is the thing, this is the simple thing you have to remember. Um, Normally for one gene, there will be two alleles. For example, capital T, small t for height, right? If one gene is having more than two alleles, it's said to be the multiple alleles condition. Understood? Uh, and again, one more thing you have to remember, if let it be any number of uh, alleles present, for example 40, 50, 60, let it be any some x number of alleles, but only two will be there, uh, well, only two will be present to form that. Uh, for example, for your simple understanding, I am saying, let it be, and self incompatibility, I will explain you that very carefully and neatly. Okay. Well, let's go into the topic self incompatibility in plants. The self incompatibility in plants is governed by gene. Yes, yes, gene will be governed by this thing. So, in this S gene, there will be 60 alleles. S1, S2, S3, like that till S60 will be there. Now, I am explaining this point to you here. Let it be any number of alleles for 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 60, right? Only two will be there and form that particular thing in that allele. For example, S1, S2, or S1, S3, S1, S4, S2, S3, like that any particular two will form that particular allele position. Okay. Let's come into self incompatibility. There are in S gene will be controlling the self incompatibility, and there are 60 alleles present in that. Out of this 260, only two will be present in that particular plant. Have you understood? Okay. Now let's understand self incompatibility with a diagram. These are anthers. This is anthers. Right? The pollen will have this self incompatibility S gene, right? For example, this is having S1 and S2. And if this falls on the stigma, the same pollen of the same plant, so this stigma will have, sorry, this pollen will have S1 and S2 genes. This stigma, right? Then what's the use of cross pollination? So to promote this cross pollination, if the pollen and the plant is having same type of genes, it won't germinate, right? It simply means if the same allele from the same plant, then it won't fertilize, right? If pollen comes from same plant, then it won't fertilize. It's called as self-incompatibility. For example, S1 S2 crossed with S1 S2, it is called as incompatible. This is fully incompatible. For example, here S1 S2 crossed again with S1 S2. For example, S1 S2 is crossed with S1 and S3. These are partially compatible, right? Because S1 gene is common, this S2 and S2, these are two different genes. So this is partially compatible. For example, S1 S2, S2 S4. This is also partially compatible, right? Because S2 is common here, right? Now S1 S2 crossed with S3 S4. This is fully compatible, right? Have you understood? These multiple alleles, some note points you have to remember. These multiple alleles always belongs to the same locus in a chromosome, right? One allele is present at a locus at a time in a chromosome. Multiple alleles always control the same character of an individual, right? However, the phenotypic expression of the characters will depend on the alleles types of allele present. For example, this S1 to S60 genes will have same character, right? The phenotypic expression may change. For example, ABO blood group. In this IA, IB, and IO, these three will be having same character, that is blood group. However, the phenotypic character means A blood group or B blood group or O blood group will be depending on the whatever the alleles that are present. Okay. And the next point you have to remember is there is no crossing over in multiple allele series. In, for example, the wild type is always dominant over the mutant type, right? Whatever the wild type is there, that is always dominant compared to the mutant type. And another thing is multiple alleles do not show complementation. What is complementation? Uh, we will discuss in the further classes where the cis and trans positions will be there. That complementation. For example, I will simply say here and we will discuss it detailedly later. Complementation test means in from this test we can find that two genes are on the same chromosome or different chromosome. With this, we will find it. For example, in Drosophila, the pigment, based on the pigment, red eye color or white eye color is observed based on the complementation test. We'll discuss it later. Mm, this is the thing. Uh, for example, next topic is pseudo. As the name suggests that pseudo means false allele, right? What actually this is? Okay. For example, in a chromosome, this is the locus where only one allele will be present, right? If the locus is very complex, for example, A and B, this is both. This both is called as one locus only with subloci. These are subloci. These are functionally same and closely linked, right? Whatever this type of things are there, means whatever they are closely linked and functionally related genes are said to be pseudo allele. It governs different expression of same character. For example, uh, we'll discuss it later. Uh, it occupies a complex locus which is divided into subloci. They have occupied different position but on same complex locus. Different position but on same complex locus, right? These are exhibiting low crossing over frequency, right? Hmm. This also exhibits cis and trans position effect in complementation. Example: lozenge I and star asteroid I in Drosophila. In Drosophila. Okay. Now we will discuss what actually is cytoplasmic inheritance. What cytoplasmic inheritance means? It is due to the DNA that is present in the mitochondria. This is nucleus inside the nucleus. This is called nuclear DNA. Whatever the DNA present in the cytoplasm. You may get one question. What DNA is present inside the cytoplasm? Yes, some organelles like mitochondria and chloroplast, which are present inside the cytoplasm. Uh, they are having its own DNA, so they are semi-autonomous body. So if any kind of inheritance that is governed by the cytoplasmic genes, DNA, then it is said to be the cytoplasmic inheritance. This is also known as extranuclear inheritance or extrachromosomal inheritance. Just remember these two other names also. Question may be asked in any way. Extranuclear inheritance or extrachromosomal inheritance. Right? Whatever the gene that is governed by the cytoplasmic inheritance are known as plasma genes. This is very important type of question. Whatever the inheritance that is gene governing the cytoplasmic inheritance, they are said to be the plasma genes or cytoplasmic genes. Okay, these are also like the characters of one progeny. Usually, the female parent are transmitted to the progeny, and this is said to be the uniparental inheritance. For example, this is male, this is female. Normally, whatever the F1 we are getting in cytoplasmic inheritance, whatever the maternal character, mother character, because whatever the cytoplasm we are getting, it's all from the mother side only, right? That's why it's called maternal uh, character. So, whatever the characters, female characters that are transmitting to the progeny, it's said to be maternal character transmission, and it's also called as uniparental inheritance. The reciprocal crosses exhibit consistent differences for such traits, right? For example, if you have changed this male parent as a female parent and female parent as a male parent, what like you may get confused like how we can change male parent into female, female into male. That is simple. If we are taking pollen from this in the first case, now we will take pollen from this parent, right? Parent A and parent B, let it be. For example, in the first case, you are taking pollen from parent B. Now you will take the pollen from parent A. Like this, if we change, this reciprocal cross may exhibit different traits, right? Because of cytoplasmic genes that are present only in the mother. So these genes are governed by the maternal characters. Okay. So the reciprocal crosses going to be B cross A. Okay, these reciprocal crosses will exhibit consistent differences for this type of traits which are governed by the cytoplasm, and segregation is not observed in F2 and subsequent generations because whatever the F1 is there, the same on selfing, same cytoplasmic genes will go for this F2 generations also, right? So there won't be any segregation observed in this F2 and subsequent population generations. Okay, it always generally it shows somatic segregation during mitosis, uh, and the sum total of cytoplasmic genes is known as plasmon, whereas this plastron denotes cytoplasmic genes and chondrium denotes the mitochondrial genes. As we have discussed in the earlier, uh, whatever the genes that are present in the cytoplasm, it is called as plasmon. And again in this plasmon, there will be mitochondrial genes and chloroplast genes. Whatever these mitochondrial genes, these are called as chondron, chondrion, and whatever this chloroplast, these are called plastron, right? Whatever the individuals which are heterozygous for plasmogenes, these plasmon genes are known as, sorry, uh, whatever the individuals with heterozygous for this plasmon genes are called as, heterozygous ones are called as cytoheads, right? The cytoplasmic male sterility in plants is controlled by mitochondrial genome, which is chondrion, right? This shows this cytoplasmic inheritance. For these plasma genes, the potent mutagen is ethidium bromide. Okay. Now we'll discuss about the example of mitochondrial inheritance. For example, cytoplasmic male sterility uh, is the example of mitochondrial cytoplasmic inheritance. In humans, there are there is some disease, LHON, which is called as Leber's hereditary optic neuropathy, and the person's bone marrow pancreas syndrome uh, are due to this mutation in this mitochondrial DNA. And one more thing, now the example of this plastid inheritance, what actually it means, leaf variation in Mirabilis jalapa is due to the female parent, that is from this plastid inheritance, and mitochondria is lost in the male gametes during gamete development, so it is uniparental inheritance. And one more thing you have to uh, like remember, promiscuous DNA. There is a term called promiscuous DNA. This simply means the migration of DNA molecules from one organelle to another organelle within the cells of the eukaryotic tissue. Okay, usually one DNA or one gene moves from chloroplast to mitochondria. From chloroplast to mitochondria, it will move. Okay, into the nuclear genomes. This type of thing is said to be promiscuous DNA. Thank you.